Well, hello everyone. My name is Michel. Thank you very much for joining us. Today, I am really delighted to host Professor Sonia Gandhi, who will be talking to us about finding the energy, what happens to mitochondria in Parkinson's disease. Now, I guess that if you're joining us today, you probably know that mitochondria are known as the powerhouses of the cell, and they produce the energy necessary for the cell's survival and functioning. So Professor Gandhi is an expert in this area, and she established a laboratory at UCL, at the UCL Institute of Neurology in 2013, and was awarded the laboratory secondment to the Francis Crick Institute in 2016. As always, I would like to remind everyone that this session is for information and education purposes only, but you know that if you are seeking medical advice, this is not the right forum. Please make sure that you consult a medical professional. As always, there will be time for questions at the end of the presentation. So if you want to ask a question, please use the Q&A function at the bottom of your screen. Do not use the chat, use the Q&A function, please. So for those of you who don't know us yet, No Silver Bullet is managed by Mark Lambert and myself with the aim of sharing Parkinson's expertise. We aim to help you and frankly to motivate you to make informed choices on how to adapt your lifestyle, to manage the symptoms, and hopefully to slow down disease progression. We post the recordings of our webinars on YouTube. We reached 10,000 subscribers today, so thank you very much for your support. And on Spotify. And we also post short videos on TikTok and Instagram twice a week. The details are available in the chat section at the bottom of your screen, as well as on our website. And our website is, no surprise there, nosilverbullet4pd.com, nosilverbullet number 4pd.com. But let's come back to today's topic and to Professor Sonia Gandhi, who will be talking to us about what happens to mitochondria in Parkinson's disease. Sonia, the stage is yours.
Well, thank you. Thank you so much for having me. And I'm going to share my screen now. Is that okay, Michelle?
Perfectly fine. Yes, go ahead.
Okay.
Perfect.
So I'm a clinician scientist, which means that I see patients with neurological conditions, including Parkinson's, and I also run a research program at the Francis Crick Institute, which is to try to understand the causes and what's happening inside the brain of people with Parkinson's. And that's interesting. There we go. So let me tell you a little bit about the beginnings of mitochondria, really just to set the scene of what I'm going to tell you about their involvement in this condition. So really, we date back mitochondria about 1.5 billion years ago. And 1.5 billion years ago, an event happened, a kind of once in an evolution event. And in that, mitochondria weren't mitochondria then, they were these primitive bacteria type organisms. And a certain cell that was existing around that time took it up, just ingested it. And normally, when a cell might do that, it would digest it and throw back out the contents. But in this one event that happened, what actually happened is that that bacteria learned to live within inside a cell and learned to adapt to the host cell, which you're seeing just here, and provide energy for that cell. And from this cell came all animal, or what we call eukaryotic cells after that. And that's really, really important because the fact that this small bacterium that then later became known as the mitochondrion within a host cell, once that event had occurred, it meant that that host cell could produce energy, it could increase in size, it could change its complexity, and eventually it could give rise to the sort of complexity that we see in mammals and humans. And that event also happened with something called a chloroplast for plant cells, giving rise eventually to plant life. So it was really important that mitochondria came into eukaryotic evolution. But why does the brain need mitochondria? Another massive evolutionary step was the development of the brain. And as you've already heard, the main answer to that is energy. So if you think about the human brain has about 100 billion cells or neurons or nerve cells. Each neuron will have about 10,000 connections. That's about 1,000 trillion synapses or connections between neurons in networks. So that is a huge, huge computer. And although the brain only weighs about 2% of the body's weight, it in fact uses about 20% of the whole body's energy requirements. You know, and it's actually very, very energy efficient. So if you think that to make about 1 billion computations per second, the brain uses maybe about 12 watts of power. And you can compare that to a sort of a supercomputer that can do a simple language model in AI-based terms, and that uses several million times more energy. So actually, the human brain is very energy efficient, but of course, it needs to get that energy from somewhere. And what I'm showing you here is a diagram of the mitochondria then, where most of that energy is being generated. And on the left, you're seeing a healthy mitochondria. And through a series of chemical reactions that take place in the inner mitochondrial membrane here, these folds or cristae, there is a series of chemical reactions which allows energy to be then stored as a fuel, and that fuel we call ATP, and then that fuel can be used to release energy where it's needed in the cell. Now, the whole process of producing energy, though, is also has side effects. And one of those side effects is to produce reactive oxygen species, which are free radicals that are generated when energy is generated. Now, what they can do is go on to damage components of the cell, proteins and lipids and DNA. And that can cause problems. And those problems are apparent in a range of different neurological conditions that I've just listed here. And so the cell has adapted many ways to both make energy, but also to deal with the sort of side effects, if you like, of producing energy as well. But now coming to the sorts of cells that get affected in Parkinson's, we know that one of the earliest cell type to be affected are the dopaminergic neurons, the cells, the brain cells that are producing this chemical transmitter called dopamine. And that results in a relative loss of dopamine in the brain. Now, dopaminergic neurons, interestingly, what I'm showing you here is a cartoon of the brain, and all of these groups of red and yellow are the nerve cells that are affected in Parkinson's, and they all share a very specific characteristic, which is that their architecture is very unique and very, very complex. So this is a single neuron, and what you're seeing is that this would project from the brainstem through to the striatum and would secrete dopamine. But you can see this very big arborization is what we call it, because it looks a bit like a tree, and it's incredibly complex. Now, in the brain, the larger a structure and the more complex a structure, the more mitochondria it needs. So it needs batteries along all of these dendrites and all of these axons. So one of the issues is that neurons, especially neurons that make dopamine, are very complex. They have this very unique architecture, and therefore they need a lot of energy. And that makes them very vulnerable then if the mitochondria are not working properly. So that is just to set the scene of why the brain is so dependent on the mitochondria. But of course, as I think about how we can treat Parkinson's, what we really need to do is understand what underlies it. Now, our understanding of Parkinson's has changed really dramatically over the centuries, really, and some of the earliest accounts, which date back to the Sanskrit literature, describe tremor and slowness of movement. And then more in the Western literature, and of course, the very famous essay on the shaking palsy by James Parkinson describes a predominantly movement disorder. But really, our understanding has changed much more in recent years, that we understand the condition to affect the motor circuits of the brain, but also the non-motor ones, and to cause a really large array of different symptoms. And we often think then of the symptoms presenting potentially with the movement problem. So I might see a patient here at time 0 when they come to me with tremor or rigidity or bradykinesia and slowing of the movements. But we'll know that their condition will have been present for many years prior to that, with symptoms such as constipation or lack of smell. or sleep problems. And over the years that I'll look after people, I'll also see their condition change as they develop other different symptoms as different parts of the brain are affected. And in this phase, we know that this is mainly due to a lack of the dopamine neurons, the ones that I've already shown you are very complex in the human brain. And so when we want to understand therapies, we need to understand what is happening in this phase in the dopamine neurons so that we could try to slow down conversion to the motor syndrome. And we also need to understand what's happening in this phase during progression throughout the rest of the brain in other neuronal subtypes to try to slow down non-motor progression. So understanding those mechanisms is really, really critical to a researcher trying to find new ways of treating the condition. So what do we understand so far? Well, approximately 25 years ago, we probably didn't understand that much about what the actual molecules or genes or proteins that were involved in Parkinson's were. But cueing genetics and better forms of sequencing the DNA, we've understood a lot more. So we know that people with familial forms, inherited forms of Parkinson's, which make up a real minority, somewhere between 5 to 10% of people, may have a mutation or an abnormality in one of these genes shown here in red. And that puts them at much higher risk of Parkinson's, but they're incredibly rare. Whereas all of these genes just shown here in green are risk factors. They're risk little SNPs or variation in the human DNA that puts us at a little bit more risk of having the condition. And these are much, much more common. And it's these that really cause what we would call idiopathic or sporadic Parkinson's. But what we know from these genetics is that all of these genes map onto only a few pathways in the cell. And I've just shown them here, the way in which we deal with misfolded proteins, our immune system, but really importantly, the mitochondria as well. So we know now genetically that there is a chance that the way our mitochondria work may predispose people to getting Parkinson's. Now, if that's the genetic risk, we also know that there's something on the other side of this that I'm going to call the environmental risk, because it's a condition where the genes that we're inheriting really interplay with the environment. And what is it about the environment? Well, there's a number of factors we know contribute, and they include, for example, the way our brain is aging, exposure to toxins in the environment, exposure to viral infections, and other lifestyle measures such as exercise and diet. So where does mitochondria fit in with all of this? Well, if we think of one of those main factors as being brain aging, we know quite a lot that as the brain ages, a number of changes take place in the human brain, and they affect the communication between neurons. The DNA becomes less stable. We don't repair proteins very efficiently as we age, and our cells go through something called senescence, which is an aging phenotype where they don't function quite as well. But critically, our mitochondrial function, these batteries, they get tired as we age, they age as well, and they don't function as well. And actually, in the mitochondrial theory of aging, it is often described that once the mitochondria start to decline, that actually accounts for all of these other aging phenomena that we can see in the brain. And so actually, if we could understand why the mitochondria contribute to brain aging, we would then also understand why brain aging contributes to conditions such as Parkinson's. Well, I talked about brain aging, but let me tell you about that other risk factor, the environment, and how mitochondria play a role in that. And this is really a puzzle. The first piece of understanding the puzzle of Parkinson's came back in the late 1970s, early 1980s. And I'm just going to describe what actually happened. So this was based in California, and a group of approximately 6 individuals who were young had developed a form of Parkinsonism. And I say Parkinsonism because they'd become rigid and frozen or with tremor. And that was quite unexpected because they were aged sort of 21, 30, 35. And what connected them was that they had all been taking illicit drugs and they were trying to inject heroin, but in fact they were all linked together because they were using the same batch of drugs. And in fact, it wasn't heroin, it was somebody synthesizing an opiate in their own laboratory, in sort of a homemade laboratory. And when that substance went off for analysis to the lab, what was found was that it was contaminated or laced accidentally by a toxin, something called MPTP. And what had happened is that the MPTP that these individuals had inadvertently exposed themselves to entered and crossed the blood-brain barrier. And then it had gone into something called the glial cells, where it had been metabolized to something called MPP+. Now, MPP+ went into a dopaminergic neuron through a transporter that we call the DAT transporter. And there it gone into the mitochondria and it had poisoned a complex called complex 1. And these are the complexes that make the energy in the mitochondria. So then these mitochondria could no longer produce energy. And as a result, these neurons, the dopamine neurons, died in these individuals, and they developed what looked like Parkinson's disease. And in the first few years after this discovery was made, they were treated with levodopa therapies and had a partial response. And 2 of them went on to have a stem cell transplant in Sweden, and another went on to have some deep brain stimulation. So they didn't have the completely typical Parkinson's, but they had a form of it. And it was the first proof that a toxin that inhibited the mitochondria, that stopped mitochondria working, could cause Parkinson's. And before that, we had no idea what a cause could be. So it was a really pivotal moment in understanding the condition. So it told us that environmental toxins could inhibit mitochondria, and that in turn could cause Parkinson's. And those toxins we know now are not just MPTP, which was this inadvertent, accidentally discovered one, but also pesticides such as rotenone and paraquat. But also for a researcher, it allowed us to make new models for Parkinson's disease because we didn't have any models before that. So if we wanted to see in a mouse model or a cell model or a primate model how to study the condition, we had no way of making it. But now we could give these toxins in a laboratory environment and generate a model that could then be studied. So this was probably the first piece of the puzzle, that mitochondria really were going to be important. Well, the second piece then comes from our understanding of genetics. As I've already said, we've got some understanding of the actual molecules that underlie the condition now, just from the last 25 years. So in 2004, a family was discovered, and they have early onset, what we call recessive Parkinson's, and everyone in black here is an individual affected. And when the lab went on to sequence the DNA, they found a mutation just here in a gene called the PINK1 gene. Now, early onset Parkinson's in this family was due to a mutation in the PINK1 gene, but actually mutations in these other genes, Parkin, DJ1, FBOX7, that can also cause autosomal recessive PD, encode proteins that go to the mitochondria. So that was very interesting because now we knew that if a protein in the mitochondria was upset or mutated or abnormal, that would lead to the condition that we understand as Parkinson's. Now, these forms of Parkinson's aren't actually completely typical. They form a sort of subgroup of their own, where they often will be early onset, under the age of 40, but even at the ages of 20 and 30. They tend to be very, very responsive to levodopa, but there are motor fluctuations that happen despite treatment. And sometimes they also present with muscle cramping or dystonia of a limb at the beginning. So they're similar to sporadic Parkinson's, but not entirely typical. But clearly the mitochondria is the culprit for these conditions. Well, this was back at the time that I was just doing my PhD in neuroscience, and after the discovery of PINK1, we set about trying to generate neurons in a dish and look at the mitochondria, and they're painted here in red. This is what neurons look like in a cell, and in red you can see what the mitochondria look like. And what we went on to show, these are other brain cells, these are glial cells, that when the cell takes up calcium, shown here in green, the mitochondria shown here in red also take up that calcium and become very, very abnormal. And you can just see the green and red overlapping here in these cells. So we showed that actually in people who have a mutation in the PINK1 gene, but also in the Parkin gene, they really have abnormal mitochondria sitting in these neurons. But in fact, the damaged mitochondria, they're not generating much energy, but actually these genes are doing something else as well. What they're doing is when mitochondria are damaged by aging or by the environment or by a toxin, mitochondria accumulate and get labeled with something called ubiquitin, these small little chains of UB. And then PINK1 comes and sits at the outer membrane of the mitochondria and triggers Parkin, another gene that causes Parkinson's, to be recruited to the mitochondria. And then the mitochondria that are damaged get decorated with all of these ubiquitin chains, and then recruitment of a disposable mechanism called the autophagosome happens, and then damaged mitochondria are removed and new mitochondria are made. And this process we call mitophagy is a sort of quality control process, and it's absolutely fundamental for neurons that live for 80, 85 years, your whole life, you're born with the same neurons, that those mitochondria can be cleared when they're damaged and new ones can be made. But in Parkinson's, this process, and particularly in Parkinson's with these gene mutations, this process doesn't work very well. So we started to understand that not only are mitochondria important because they produce energy, but actually when damaged mitochondria are not cleared, that in itself is enough to trigger the condition. Now, after discovering these genes, we also found out another problem that can happen when damaged mitochondria occur. And what I'm showing you here is a schematic where here's the mitochondria, and you can see PINK1, Parkin, all these genes that cause Parkinson's are shown here, and as well as an energy problem that happens. The other thing that can happen is that little holes get poked in the mitochondria. So mitochondrial contents, including DNA that exist in the mitochondria, as well as mitochondrial sort of death molecules and signaling molecules, they all can get released from the cell and leave the neuron. So surrounding the neuron in the brain are a host of immune cells, microglia, astrocytes, and they're there to really patrol the brain and also to support neurons. But when mitochondria get damaged and release their contents, they get activated and induce inflammation. So the other problem with damaged mitochondria is not just that they don't produce the right amount of energy or that they can accumulate, but actually they in themselves can become a little bit toxic by inducing inflammation. So there are many ways then in which mitochondria can affect neurons. Now, we talked about sort of environmental forms of Parkinson's and then these weird genetic forms or these rare genetic forms. But what about common Parkinson's? So common Parkinson's is really identified by the fact that there is protein misfolding. So there can be rare mutations in genes that encode alpha-synuclein and LRRK2 and tau, but in fact, the commonest form is not associated with mutations in genes. It's associated just with a protein called alpha-synuclein that misfolds and is deposited as what we call a Lewy body. So if most of Parkinson's involves protein aggregation and some forms of Parkinson's involve mitochondrial dysfunction, what I've spent really most of my laboratory career has been trying to understand how they're interlinked and why is it that both processes can occur and which one comes first. And can we account for the whole spectrum of the condition, where people can progress very slowly and the condition can stay in the brainstem, versus people whose condition can spread throughout the brain using both protein aggregation and mitochondrial dysfunction. So the way I do that then is to make models of human brain cells in a dish. And we're able to do that because we can take skin cells from individuals who have mutations in these various genes. We can then turn those skin cells into stem cells. That's a Nobel Prize-winning technology, which allows us to take a skin cell, reprogramming it, turn it into a stem cell roughly around the age of a fetus, and that stem cell is capable of becoming any cell in the body if we give it the right cues. It's a bit like giving it sort of postcodes. So we turn the stem cell into a cell that is part of the nervous system, and then into a part of the brain that's affected in Parkinson, such as the midbrain or the cortex, or into one of these glial cells. And this is what they look like in a dish when we've painted them with specific cell markers. So now I have quite a good model in a dish of my patient's brain cells. And the next question I wanted to ask was, well, where do these protein aggregates form inside a cell? We know they're there. We've seen them in brain tissue for many years now, since they were first described. And this is what they look like, these Lewy bodies or Lewy neurites. And they're quite big, actually. They're several micrometers, so you can see them with a standard microscope. But where do they come from? Well, they actually come from the protein, which is only nanometers in size, beginning to misfold. And as we sit here, 1% of our protein is misfolding all the time, but our cells are really good at refolding them and making them better and right again. So if a bit of our proteins are misfolding all the time, some of them will go on to form bigger protein aggregates called oligomers, and those will change their structure and they'll eventually form Lewy bodies. And our aim is to try and understand these ones and where they're forming inside our human cell models. So what did we find? Well, to our surprise, when we made human neurons from the front of the brain, the cortex of the brain, and then we added alpha-synuclein to those cells, so that's the color that you're seeing here. And then what we did was perform electron microscopy, which is the most ultrastructural way that we can look inside a cell. So we're diving into a human neuron here. This large thing you're seeing is a nucleus, and all these small things are mitochondria. And we're asking, where does the alpha-synuclein go and where does it begin to form aggregates? And you can see these little colored circles here are the aggregates forming. And we can look at using the EM at what structures they're forming on. And then after 7 days, we can see more like Lewy body-like structures. And again, we can see what structures they're on. And we were quite surprised to find that actually the mitochondria was one of the locations. There are many locations, but it was one place in the cell where these proteins can start to misfold. And then we tagged the mitochondria using a green tag called MitoGFP. And you can see the mitochondria here in the electron microscope, and the alpha-synuclein in red, and in yellow is where they overlap. So we then set about making little vesicles made of lipids, made of the mitochondrial membranes, and added the alpha-synuclein and showed these aggregates forming. And in fact, when you take the aggregates, this is what they look like. They form these fibril structures. This is just the protein that makes up the Lewy body, and they can form on mitochondrial membranes. Well, that was really important because now we had a link between this common process that occurs in everybody with Parkinson's and the mitochondria. And we described in this piece of work now that actually alpha-synuclein needs a sort of surface in which it can begin to aggregate on, and that could be the mitochondria shown here. And that's really important because in the mitochondria, it can then poke these little holes and cause lots of chaos. So it can reduce the amount of energy that's produced, and it can generate a lot of these free radicals that go on to damage other structures in the cell. And it can go on to induce cell death. And we'd shown previously that just normal alpha-synuclein doesn't do that. But when these little aggregates form, either at the mitochondrial surface or elsewhere in the cell, they then interact with the enzyme that produces the energy. It's called ATP synthase, and it pokes this big hole in the mitochondria. And just to show you what that looks like, these are cells where the mitochondria are painted red, and the cell death marker is painted green. And what you can see in the video is the red disappears out of the mitochondria, and then the death signal increases in those cells. So that process happens when we add these little aggregates to neurons. Well, I've explained to you then that mitochondria are important in environmental forms of Parkinson's. They're also important in the rare genetic forms, but now they're important in the common form where all proteins misfold. But what comes first is a question that we've been asking for a long time, and it's really difficult to understand because we can't study the brain in life very easily other than via things like MRI scans. So what we went on to do then was to go to a brain bank that we work with and take postmortem brain tissue and take it from people who had passed away at a point in time when their Parkinson's only affected a small part of their brain. But what we did was sample all of the brain structures and ask what is happening there. So before the brain develops Lewy bodies, before you see any neurons dying, what is actually going wrong? And again, to our surprise, we did this by measuring thousands and thousands of proteins in all of these different regions. But the top pathways that came out in all of these regions that don't yet have the Parkinson's pathology were that their mitochondria weren't working properly, and that oxidative phosphorylation, which is the way in which the mitochondria produce energy, wasn't working properly. And sirtuin signaling, which is the mitochondrial basis of aging, also wasn't working properly. So that changed the way we thought about the mitochondria, that's not happening late in the condition, that in fact what happens is that the brain goes into a stressed state where it can't meet its bioenergetic demands because mitochondrial decline is happening early. And then that provides this sort of environment that makes vulnerable neurons start to misfold proteins. And therefore you have this sort of perfect storm, if you like, of events happening where you've got brain aging and the mitochondria not working so well at the same time as the mitochondria declining because maybe those mitochondria weren't so functioning well, and then the misfolding of the protein might occur, positioning mitochondrial dysfunction really early in the condition, even probably long before symptoms develop as well. So what I've told you so far, just to summarize, is that actually mitochondria are important, but through many mechanisms, partly through energy, a lot through energy, but also to do with the way that they sort out the stress in the rest of the cell and causing inflammation. That we have found mitochondrial dysfunction in many models in the laboratory, the environment models, the genetic models, but also when we try to look at sporadic Parkinson's, the common form of Parkinson's, that we think it occurs early in the condition, and that mitochondria may provide this link between brain aging and vulnerability to the condition. So in the last few minutes, what I'm going to talk about is, can we change anything? What can we do to improve mitochondrial health? And I've taken this figure from this really excellent review because I think it summarizes everything that can conceptually be done, because it thinks about it as a top-down approach as well as a bottom-up. So top-down, thinking of the whole human. Now, we know that exercise and good diet is really, really helpful for mitochondria in our muscles. So we know that exercise will improve mitochondrial function. It also produces more mitochondria in muscles. We actually don't know very much about how exercise changes the mitochondria in the brain. And that's because we're not very good at assessing mitochondria in the brain during life, which is a problem for us in the research community. There are various approaches that we use, such as deep brain stimulation, that people are speculating may change mitochondrial function as well, but I would say that we don't know that much about that yet. If we look at cells in culture, which is what I'd spent a lot of time doing, we know that there are many ways to boost the way in which mitochondrial metabolism work. A lot of supplements that can be taken that boost the energy production, and small molecules as well. Or we can try to target mitochondria themselves, which is very difficult to do. Or we can try to circumvent the effects of the mitochondrial dysfunction. So where we know that abnormal mitochondria cause more protein aggregation, can we try to use antibody therapies to remove those protein aggregates? We know that abnormal mitochondria result in inflammation, so can we use anti-inflammatory approaches? We know that we induce oxidative stress, all those free radicals that happen and damage other parts of the cell. So can we use antioxidants? So these are sort of mechanisms and processes that are trying to circumvent mitochondrial damage rather than targeting mitochondria themselves. So those are sort of conceptually what we can try to do and what is actually being done. So this is from an excellent review by Kevin McFarthing, who writes about and summarizes all the current clinical trials that are happening as of this review is as of the 31st of January 2024. And what you're seeing here on this side are all of the trials for disease-modifying therapies and all of the trials for symptomatic therapies. Now, of course, everything I've talked about is trying to target mitochondria because we want to find a disease-modifying therapy. And so you'll see these ones in orange here are the current trials, so that are targeting energy in the mitochondria. There are about 4, which makes up about 7% of the trials currently. So there are trials starting, but actually many of the things that are really happening in the community, or even before that, they're happening still in the laboratory, where some of these events that I've talked about today, changing mitochondrial quality control, reducing inflammation, stopping this lipid interacting with the alpha-synuclein, are all happening still in preclinical development before clinical trials. But just to spotlight a few of those trials that are going through. So this was a trial of ursodeoxycholic acid, UDCA. And UDCA is a small molecule that rescues the mitochondria in models in the lab. And in this study that was done in the UK, they took people with Parkinson's with an early diagnosis and gave them either a placebo or UDCA. And then what they did was measure, really the aim of the study was for safety and tolerability, which they showed it was safe and tolerable. They did look at progression of the motor symptoms, but really the trial is not sort of designed to do that at this sort of small number. But then they went on to look at the brain using a special brain scan that measures, or at least captures, the energy levels, the ATP levels. It's called magnetic resonance spectroscopy, and it's a type of MRI type scan. And that hadn't been used before in a trial for a mitochondrial therapy. And what they did show was that UDCA did improve the mitochondrial function in the brain. And so this is moving forwards to the next stage trial to really test it on a much larger number of patients to see if it's effective in slowing down progression. And the second one that I think is also gaining in importance and will become one that you may have already heard about is a supplement, which is a vitamin B3 supplement. And what that does is the nicotinamide riboside actually supplements one of the substrates that's needed for the complexes of the mitochondria to work. And so it boosts energy production. And actually, when we give that supplement to people in these really early trials, you can then measure the NAD levels actually peripherally and show that it indeed has had its effect in improving mitochondrial function. And this again has gone through phase 1 trials and is moving through to phase 2 and then phase 3 trials. So we're yet to understand whether these mitochondrial therapies will change progression, but they are very, very active, and they're the two that I think are particularly important to look out for. There's one other strategy that's being used as well to try to improve mitochondrial function. And these are this selection of diabetes or weight loss drugs called the GLP-1 receptor agonists. And the glucagon-like peptide 1 receptor is important because we know that while patients with diabetes have insulin resistance in their periphery, they also have insulin resistance potentially in their brain, where insulin plays a really important role in protecting neurons, but also in mitochondrial function. And when we give these drugs, like exenatide, for example, into cells in culture, we find it really boosts mitochondrial function. And so this was a phase 2 trial performed at UCL, where exenatide was given once weekly over 2 years. And what you're seeing here is the exenatide treated group improved at the start, maintained an improvement. This is in their motor scale part 3, which is a scale we use in trials, whereas the people on placebo progressed slowly over that period of time. And so we're awaiting the results now of the phase 3 trial that's been conducted to see if these GLP-1 receptor agonists that we think also operate by improving mitochondrial function, but also by reducing inflammation, whether that will be successful. And then you may have also heard of another GLP-1 receptor agonist called lixisenatide that was recently reported in the New England Journal. And this took people with Parkinson's diagnosed within 3 years and either gave them the GLP-1 receptor agonist or a placebo. And what they showed was that the patients with placebo progressed by 3 points on this motor scale, whereas those who'd been treated with this GLP-1 receptor agonist didn't progress during that time. And much longer term follow-up is needed, and indeed larger trials are needed for both of these medications, but they're also encouraging as another way to improve mitochondrial function. So with that, I'm going to just summarize what I hopefully have told you, which is that I think mitochondria really are critical to the function of neurons, both in the resting brain, in the aging brain, and certainly in the condition Parkinson's. That dysfunction of the mitochondria may be acting directly, a deficit of energy, or indirectly by setting the cell into a cellular stress state or the brain into a stress state. And there are then many overlapping mechanisms where a stressed brain then undergoes protein aggregation that worsens the mitochondrial function. We certainly need really good ways to measure brain mitochondrial health during life, because at the moment, all of our measures of the mitochondria are in the periphery outside the brain, and it's really hard to assess it inside the brain. That if we could find ways to maintain mitochondrial health, or even boost mitochondria, we might even be able to protect the brain prior to the condition starting. The idea of preventing motor conversion or slowing down progression becomes really, really important here. And then there are these mitochondrial-directed therapies that we are hopeful may slow down progression. And we think they'll be doing that by protecting the vulnerable neuronal subtypes really at the time that they are still functioning, but can be boosted, and their function can be enhanced by mitochondrial-directed therapies. And with that, I'm going to stop talking and just acknowledge all the people who fund my research, and very much all the participants and patients and donors who contributed cells to the lab for research, brain tissue, for all of the analyses that have been done by us and many others, as well as all the people who fund, support, and collaborate with us. So thank you very much for listening.
Sonia, thank you very much to you for a great presentation. Can you please stop sharing the screen? Thank you very much. Because you've been tackling a very important and complex topic and making it understandable to hopefully all of us. I just wanted to mention to our audience that if you want to know more about lixisenatide, we have on our YouTube channel a presentation by Professor Oliver Raskol about the topic. And we also have concerning UDCA that Sonia mentioned, Professor Oliver Bandman presenting at one of the Cure Parkinson's research updates. So you can find half-an-hour presentation on each topic to your delight. Let's just look at the questions that we have already coming in here. I think that Talib, I will probably handle that one. In one of your charts, you were showing exercise in the same half triangle that environmental factors. And actually, I don't think you meant for a second that exercise was a risk factor, because you spoke after that about the benefits of exercise.
Yeah, just to clarify that, I really mean exercise is one of the sort of modifiable environmental factors, but it's the sedentary lifestyle that is the risk factor, while the increased exercise is the protective one.
Thank you very much. So Jill is asking about food sensitivities and whether those could be helped by stimulating mitochondria with red light therapy, photobiomodulation. And if so, how would one avoid weight loss? So there are two topics there embedded in one.
Yes, gosh, okay. So, you know, I mean, I think I will start by saying that I'm not entirely sure how we understand the way mitochondria work, for example, in the gut, compared to the way mitochondria work in the brain. So everything I've talked about has been very, very much about brain cell mitochondria. And so everything else is slightly speculative. And now I think, I mean, weight loss is a really important problem that we struggle with, and we've struggled with just the last bit of my talk on the GLP-1 receptor agonists. Our biggest problem actually in using them is that the major side effect is weight loss, and we don't want to induce more weight loss. So actually it is a real problem. I'm not sure we fully understand the mechanisms underlying that weight loss. And because we don't really understand all of that, I'm not entirely sure that we know how to target it and what the effect is going to be of modulating mitochondria on that weight loss.
Mm-hmm. Now, the question also contains, I think, a reference to photobiomodulation. Is it something, I know that there is a lack so far of trials around the world. I know that some companies are funding some trials at the moment, but based on what you know about the benefits, the potential benefits of photobiomodulation, what is your view on that as a mitochondrial support or stimulating factor?
Yeah, I just don't know enough about the
it's a tricky one. I think we need a bit more data.
Yeah, I certainly, yeah, don't feel expert enough to comment on it because I don't think we just understand it enough yet. Let's see the outcome of the study.
I understand, that's perfectly fine. A question from Pilar. Pilar is asking about the role of the female hormones, the melatonin and endocannabinoids play in mitochondrial function or dysfunction. So there are three topics there: melatonin, female hormones, and cannabis.
Yeah, I mean, cannabinoid signaling is quite complex in the brain, and I'm not sure it's directly, I don't think we understand, again, its direct effects on the mitochondria itself. Whereas what happens is with a lot of the signaling pathways, I mean, it is true also for melatonin, mitochondria may change as a result of other signaling pathways that are happening within the cell. And it's really difficult to know whether, if you give something that boosts a circuit and as a response that cell is a slightly healthier cell, that the mitochondrial function is doing it, or whether it's a byproduct of that signaling pathway, or whether it's because it's directly affected the mitochondria. And there are so many of these small molecules and compounds that we don't fully understand whether all of those receptors, where they're all sitting when we give the drug. And I think that's completely true for melatonin as well as the cannabinoids. And the use of cannabinoids.
And female hormones, yes. Sorry.
Yeah, the use of cannabinoids as well in Parkinson's is complex, and I do get a lot of people ask me as a clinician what to do, and I guess our difficulty is that the large scale trials again haven't been done, so the data is lacking, and when we don't know benefit, we've really struggled to understand mechanism then.
So there is a total lack of research on females and women and Parkinson's. Yeah. There is very little. And Pilar was asking about the role of female hormones. Do they see them interacting with the mitochondria?
Yeah. Again, I think it's so important. And we know that, again, the estrogen receptor plays, which is where we think the estrogen is acting. The signaling cascade that follows that it has a lot of signaling pathways that are stress pathways and protective pathways, and they ultimately can affect mitochondrial function, but so indirectly that we don't know whether it's a byproduct or whether it is directly from hormones. So it's just a very messy, complicated picture, I think, understanding the effect of different hormones. But I do fully agree that actually, because we don't use female and male models in quite the right way in the laboratory, we don't fully understand the effect of gender on the biology.
Yes. So I have another question from an anonymous attendee asking, how can mitochondrial dysfunction be measured clinically? You refer to the fact that it's particularly difficult with live individuals, but I don't know if you want to elaborate a bit on that topic.
Yeah, so what we might do for people who have what we call primary mitochondrial problems is you can, from their blood, take markers of mitochondria, so the mitochondrial DNA you can measure. You can take their cells, platelets, blood cells, white blood cells, and measure the mitochondrial function because those cells are still live. And you can do biochemistry assays and measure the enzymes in the mitochondria and work out, are they active, are they not active? So there's lots we can do in the periphery. Where our biggest problem is knowing whether, because of course we have mitochondria in all the cells apart from red blood cells, whether what's circulating in the blood is reflecting what's happening in the brain. Because what's circulating in the blood, of course, will reflect muscle mitochondria and cardiac mitochondria and renal mitochondria and liver mitochondria. So that's the hardest thing. So we can measure a lot about mitochondria, but we don't know if we're measuring brain mitochondria. The only way at the moment to measure brain mitochondria is using this unusual form of imaging called magnetic resonance spectroscopy, which is not very available and is really only a research tool. So if we could, that would really solve lots of problems.
Thank you. Now, there always comes a point with a well-educated audience like I have that there is a question that I will have to ask without having any clue what I'm talking about. But hopefully you do. Does NFR2 nuclear factor erythroid-2 related factor have a role in optimizing mitochondrial function? And is there any research addressing NFR2 in PD?
Yeah,
what is it?
That's a great question. That's a great question. So NRF2 is a sort of master regulator of the antioxidant response. So as I said, mitochondria make energy, but they also at the same time as making energy, produce a lot of these damaging free radicals, which are charged particles that go on to damage proteins and lipids and DNA. And so we've evolved to have a whole lot of mechanisms to control the levels of these damaging species. And NRF2 happens to be one of the regulators of the whole response of that. And there is quite a lot of work being done, by the pharmaceutical industry in developing NRF2 activators to see if we clear this oxidative stress, then we might improve mitochondrial function as well. And certainly at a cellular level, so in a dish, that it is true that NRF2 activation is highly protective to the cells and then the mitochondrial function is boosted. But whether that is true in humans in life or with the condition, again, needs to be borne out in clinical trials, but those molecules are in development.
Thank you. Someone is asking whether there is any correlation between childhood diseases such as scarlet fever or synovitis dance with the later onset of PD or Lewy body dementia.
These are great questions, by the way. Yeah.
you will feed them to PhD students afterwards, I'm sure.
So it's a very timely question because not so long ago a paper has been published that looked at UK Biobank data. Now, UK Biobank is this great initiative that has hundreds and thousands of people enrolled, followed them through life, and has a lot of information about their condition. And also has some brain imaging and has genetic data. And what those authors showed, not just from biobank data, but also from another big database in Europe, is that if the number of exposures to certain viruses does seem to be associated with an increased risk of all of the neurodegenerative conditions, so Alzheimer's as well as Parkinson's, and many causes of dementia. Now, the viruses that were implicated in that study were sometimes viruses that directly affect the brain. So, for example, some of the what we call neurotropic viruses that can cause encephalitis, they can then be a risk factor for later developing neurodegenerative conditions. But some of them were viruses that cause pneumonia as well, right? Now, in that study, none of them suggested scarlet fever in that particular study, and that's probably the biggest study that's been done to date. So I would say that we don't know for any one particular pathogen like scarlet fever, but there is a link between the number of viral infections somebody's had in life, and particularly any one that's affected the brain, and whether they go on to develop dementia later in life. It's just an association, like any other risk factor. It's not. It has a small effect, but the fact that they are linked tells us something about how. I talked a lot about the mitochondrial theory of aging, but there's another theory of aging called inflammaging, which is related to how our immune system changes in the brain over time as well, and how it can become chronically stimulated so that there is a low level of inflammation all the time. So now we're starting to understand maybe that the number of times the brain has seen a virus may somehow play into how the immune system functions as it ages, and that in turn may affect an individual's risk.
Thank you, Sonia. Now, Shafiq says he has 3 questions. I'm seeing 4. I think I will pick 2 out of fairness to other in the audience, so I can basically also give the opportunity to others. The first one is asking about the role of exercise, glucose intake, breathing, acidity in the blood, interval fasting. Can these things help us improve mitochondrial functions?
So yes to all of them. In the muscles. So we have very good evidence that exercise improves the number of mitochondria you have in your muscles. It improves their efficiency at making energy, and it also improves the way in which we clear. At one point, I talked about the damaged mitochondria accumulating, and they need to be cleared. So exercise improves clearance as well. So there's pretty good evidence for that, because you can measure mitochondrial function peripherally. So you do the intervention, the exercise, change the diet, and those definitely help. I'm going to hesitate because I don't actually know the data on intermittent fasting. So that one I'm not so sure, but exercise and glucose intake definitely do. Where we can't extrapolate entirely is that the mitochondria of the brain are unique. They are different to the mitochondria of the muscles, and different to the mitochondria of heart muscles. And so we can't be absolutely sure because we can't test this very easily. What we do know is the benefits of exercise and diet on the brain is going to be operating through many mechanisms, because you're going to improve the cardiovascular health, so the blood supply to the brain, for example. It's going to improve probably the way in which the brain clears all misfolded proteins. We think that's true. Again, we can't measure some of these things. So it will do that, as well as potentially boosting mitochondrial function, which is why for the brain specifically, we can't say all the effects of exercise are just via the mitochondria.
I understand. So the second question from Shafiq that I will pick is basically he's asking about glutathione production and whether it improves mitochondrial function. Now, I know that many people try to supplement with glutathione, and then there were questions about the ability of the body to really absorb it. But is it something that we should focus on?
So that's interesting. So glutathione is the main antioxidant that's used in the brain. So it's the main neutralizer of the effects of mitochondrial damage. And it's not actually made in neurons. It's made mainly in these sort of star-like cells, astrocytes or glial cells. So it gets made in a cell type and then gets transported into the neurons where it has its function. And in the Parkinson's brain, and in the study that I was showing you where we try to look before at the earlier stages of Parkinson's, we do see reductions in glutathione. It's as if the brain has gone into stress where there's more reactive oxygen species and there's a reduction in the defenses. Both happen at the same time, which makes the brain vulnerable. So there is a deficit of glutathione. So intuitively, supplementation, of course, sounds right. The difficulty is, as you've already said, that we don't know whether supplements reach the right place, in the same way that we don't know whether mitochondrial supplements reach the right place at the right concentration and then can be used in the right way. So we understand the pathway but what we don't understand is how to improve the supply of glutathione in the brain.
Could we look at precursors? I think, is it NAC or NAD? I can't remember exactly which one, but that could basically help the body generate its own glutathione without having to go through the supplement.
That is very interesting. I'm not aware of small molecules that do that actually, but that is an interesting idea, because boosting the production of glutathione may be certainly one strategy. Yeah.
We can keep it in mind. So something else, Martin, I'm very happy to see a question from Martin. He was one of the first speakers at one of our sessions 3 years ago. Mm-hmm. Martin is asking whether skin cells from PD patients show mitochondrial changes, and if so, why? Martin is a chemist.
Very good question as well. So if patients have the rare familial forms where they will have a mutation in a gene, okay, and if that gene is in the PINK1 gene, possibly the DJ1 or Parkin, but I don't know that data quite as well, then actually we see the mitochondrial dysfunction even in their skin cells. Now, where it gets complicated is if we take people who have the majority of Parkinson's, the sporadic form where they don't have a mutation in a gene, and look at their skin cells, well, actually, it's just very noisy data, and we're not able to tell what is coming from the Parkinson's and therefore reflecting the mitochondria in the brain, and what is coming from the fact that skin cells get mostly exposed to UV light and therefore have ongoing damage all the time. And that also affects the way mitochondria work. And so we don't know then whether we're just sampling the fact that fibroblasts or skin cells have environmental exposures, or whether it's a marker for the mitochondria in the brain. So in the common forms of Parkinson's, I think the jury is still out. But in these rare forms of mitochondrial Parkinson's, we do see mitochondrial deficits.
Thank you very much. Talib is asking whether it is possible to enroll in the exenatide study. I think it may be a bit late, but I would recommend to Talib to check the Cure Parkinson's website for more information. Nicole is asking a general question about the gut-brain connection in the formation of the Lewy bodies. Now, I just want to say that actually your next speaker in September will be Professor Philip Skeperians from Helsinki University, who is a specialist in that topic. So we'll probably will not answer that in detail. I don't know if you want to say anything actually on this topic.
I think it's brilliant you've got a speaker in on it, because it is becoming really important. But we also know that Lewy bodies don't always, or alpha-synuclein aggregation doesn't always start in the gut and move. It can also start in other parts of the brain first. So we have to understand why there is vulnerability in multiple parts of the brain. Yes. But I think the gut-brain axis is very important, and it will be useful to hear. I will listen in to that talk.
I'm looking forward to that one too. Someone is asking very general questions about what supplements should we take or add. Now, I'm not going to ask Sonia to answer this, but I would just recommend that you have a look at Laurie Mishli's research. She's a an American naturopath. We have had her as a speaker. We will probably have her again in November normally. And she has a piece of research from the start of this year where she lists all the supplements that people with Parkinson's take, and she lists as well those that people find the most useful. So it doesn't explain why, but it just gives you an overview, a wide overview. Someone anonymously is asking, why aren't the bad mitochondria cleared in the PD brain?
Yeah.
This one is for you.
Yeah, that's a great question. So in people who have a mutation in that pathway, we understand that they can't clear their damaged mitochondria. And but for everybody else, we don't fully understand why they can't. Because there are actually many pathways to clear mitochondria. I've just shown one of them actually, but there are several different pathways. So it's as if the clearance pathways for everything that goes wrong in the cell happens. So the cell doesn't clear misfolded protein very well, which goes down a set of pathways that is different to the clearance of mitochondria. They don't clear mitochondria very well. And we don't know, is that because the sensing is wrong? They can't sense the damaged mitochondria, or is it that their clearance mechanisms are just declining with age as well? So it's a great question. We don't quite know the answer. There are also therapies designed to try to improve clearance, and they're not yet in clinical trials, but they are very close to preclinical development. So let's see.
Thank you. Someone is asking about red light therapy. We covered that already. In the same question, the person is asking about lion's mane mushroom. Unless you have a strong view on that, I would just probably leave it for Laurie in November. That will be exactly up her street. Suzanne has been prescribed calcium many years ago, and she's asking whether you could say a bit more about the necessary balance that one should try to achieve.
Yeah. So the calcium that people might take orally is very different to the calcium inside cells, where it's sequestered in very specific bits of the cell, and we've got very tight regulation. So there's not really a relationship between calcium supplementation versus the calcium in the neurons, or even the calcium in the mitochondria, which again is a place where all this calcium is kept. So we can't regulate that by what we take in. We can for other calcium problems that are in the periphery, but not the ones in the brain and not the ones in the brain cells. So calcium is really important, but actually the drugs in development have not been ones to increase calcium. They have been ones to try to block the calcium channels. There was a trial of a drug called isradipine, which is a calcium channel blocker, to try to stop the calcium moving from the wrong compartment to another compartment within the cell, if that makes sense. And that's something that sort of supplementation can't help with particularly. In fact, that trial was negative, but although the trial was negative, there's still a huge interest in trying to regulate calcium shifts that are occurring within the neuron, but using channel blockers, not using calcium supplementation.
Thank you, Sonia. Talking about supplements, Brian is asking about whether you could tell us more about NAD or NAD plus that you mentioned in one of your slides. And do you recommend it as a supplement?
So interestingly, the person who ran the study and is running the study at the moment in Norway, I think, when he's asked, should everyone take these NAD supplements? He says, not yet, wait for the outcome of the trial. And I think that's because there's a very big difference between proving something versus speculating it. And at the moment, it still would be speculation until we have the data from the trial. But the reason I highlight it is because I think it does look promising. The trial looks promising as a way of boosting mitochondrial function and knowing that it is doing that. We still don't know for certain it's doing it in the brain, but we do know it's doing it. And theoretically it should work. So I suppose I'm hopeful about it. But I mean, I'm a cautious clinician who doesn't recommend taking things that we don't have yet the evidence for.
Thank you very much. Someone is asking whether there are any cases of familiar Parkinson's inherited through mitochondrial DNA. That person is mentioning that it would then be presenting itself in males, but would be inherited through the maternal line.
Yeah. Okay, so one thing I didn't mention is that we get our mitochondria from our mothers, because they are the mitochondrial DNA comes from the mitochondria that is in the egg. And therefore our mitochondria and our mitochondrial health to some degree is modulated by our maternal inheritance. Now, the mitochondrial DNA is small. It encodes only 13 of the mitochondrial proteins. The rest are still encoded by the nucleus, which comes from both parents. So mitochondrial health is, yes, partially maternal, but also can have a paternal contribution. Now, there are conditions in which the mitochondrial DNA is mutated. We call them they're called mitochondrial disorders. And although they can have some of the symptoms of Parkinson's, they're not commonly. They actually present with other problems that can include muscle weakness or myopathy, visual problems, diabetes, cataract, epilepsy. So that it can cause a range of other conditions. Now, there are, though, genes that correct mitochondrial DNA errors. One gene is called POLG, that's called twinkle, and those genes are actually inherited in the normal way, not via the maternal inheritance, and mutations in those genes can cause a very rare form of Parkinson's, but it doesn't look like Parkinson's in the typical way. So we would call it, as a clinician, Parkinsonism, and it is causing a primary problem in the mitochondria. But the gene is not sitting in the mitochondria, the gene is sitting in the nucleus. So just to make that distinction.
Thank you, Sonia. Peter is asking about methylene blue. And if you look into that as a way to reduce oxidative stress, increase mitochondrial energy, and stimulate autophagy. Fact or fiction?
Yes. So we used to trial or think about methylene blue as a treatment for sepsis when I was training as a junior doctor. And since that time, I have not even looked at the methylene blue literature. So I will do. But yeah, I understand some of the older literature does suggest that it can be very useful as an anti-inflammatory. I'm not aware of how it works on mitochondria, but yeah, something I'll have to look up.
So Paula is asking about red light therapy again. So Paula, sorry, if you were with us before, you would have heard that Sonia is not an expert in that, so I will pass on that. But I'm sure that you can ask many people. Actually, you can view a lot of videos on that topic on our YouTube channel from 3 suppliers. Gaurav is asking whether diet changes help in mitochondrial repair. Very good question.
Yeah.
And actually, sorry to interrupt you, someone else is asking, what do you mean by dietary restriction to help mitochondria? So you can tackle maybe both, kill those two birds with one stone.
So we know from the kind of animal models that calorie restriction improves lifespan, and calorie restriction is basically reduces aging phenomenon, and one of those methods will be via mitochondria, although there are several ways in which you can reduce aging. So yes, in an animal model, I have to say I would be, because of the big problem that we talked about right at the start with weight loss, actually dietary restriction is probably not what we would advise, because of the tendency towards weight loss in the condition already. We don't know directly in humans about calorie restriction, mitochondria in the brain. Again, it's just not known.
So I will mention there that Matthew Phillips, who is one of the world's specialists in dietary therapies, looked at Parkinson's a while back, 2 years ago, I think, or a year and a half ago, with a relatively small sample. But actually, we had the pleasure of having a presentation by him, and it's on our YouTube channel. And he has already committed to speaking to us again, probably next year. So that's another one there. Jackie is asking, she's basically saying that she had one of her children's umbilical cords frozen 20 years ago. And she's wondering if you see any potential for use in mitochondrial-based PD therapies. Do we still need that with the stem cells evolution? I'm not sure.
So with I guess the stem cells that are being used now are either embryonic stem cells or induced pluripotent stem cells in the transplant trials. Mm-hmm. And I'm not aware of whether umbilical cord cells are being used at all.
Okay, maybe one for Roger Barker, who is always available, but we haven't had him in a while, so maybe next year. Helen is basically having a good question here. She basically says that she would like to understand the role of stress, lack of sleep, life events, and the way they impact mitochondrial function.
Yeah, that's an excellent question. So, I think we'd originally thought that sleep is one of those major environmental factors that alter risk to memory problems and brain function. And the reason why I'm hesitating is because we'd always thought that it was related to the way in which during sleep we might be clearing misfolded proteins, which is this other part to the process that we think is happening in the Parkinson's brain, but in many disorders. So that's the way we'd always thought, but there's been some recent papers that suggest that actually that isn't entirely true. And so now I think the jury's still out about how sleep is beneficial. Is it because it's restoring the balance of protein folding, or is it through some other mechanism? Now, life events. We also know that high levels of stress, life events, increase our cortisol levels, which is one of our stress hormones. And a little bit like the answer with the estrogen, cortisol and estrogen, you know, they will affect a receptor with many, many signaling pathways in the cell. And what it does is put the entire cell into a state of stress. So not just the mitochondria, right? But a whole load of the cell will become stressed. So the way in which you traffic proteins, the way in which you clear proteins, the way in which enzymes work. So it's really difficult to pinpoint the effect of cortisol just on the mitochondria. But the stress for life events feeds into our cortisol levels, which we know is quite detrimental, but detrimental to many things, our vascular health, our brain health as well, you know, not just one thing.
We have an amazing presentation by someone called Rick Emmerich, who works for Bas Bloom, and his presentation is on the topic of stress. And I remember that he was emphasizing the importance of chronic stress, like repetitive stress, not so much as a cause of Parkinson's, according to him, but more as an event that would be uncovering or basically making Parkinson's appear sooner than later. That's available on our YouTube channel. Someone is asking about coenzyme Q10, a bit like glutathione. There were questions about efficiency, and I think there was a study that disproved its potential efficiency. I don't know if you're familiar with that.
Yeah, so coenzyme Q10's had a long history of being thought about and trialed. And in theory, of course, it should, because it improves the electron transport across the chain that generates the energy. But the clinical trials for the CoQ10 have been unsuccessful. That doesn't mean that it's not the right approach. It just means that actually targeting the mitochondria directly is very, very difficult, because you're needing your therapy to be absorbed, get into the, you know, via the gut, into the bloodstream, cross the blood-brain barrier, which is this big barrier, then go from the blood-brain barrier into the neuron, enter the mitochondria, and have its function, and not be broken down at any point along that pathway and reach it at the right concentration. And that's been the real problem with the trials to date. So we haven't understood whether that happened and it didn't work because it was the wrong thing to target, or whether all of those steps didn't work because you can't measure it. And that's why the drug didn't work in the clinical trial setting. So CoQ10 is, you know, it is being used in mitochondrial disorders, but we just don't know, again, whether it reaches the brain mitochondria and acts in the way we want it to act.
Thank you, Sonia, for clarifying this. Stephen Moto, who was one of the founding members of No Silver Bullet when we were just a handful of people having met at a seminar, is asking two questions. First of all, he's congratulating you for a brilliant presentation. I can only agree. Then he basically says, is there a point where mitochondrial health is so bad and advanced that there is no benefit in addressing the mitochondria anymore? Is there like a point of no return when everything has been spoiled, so to speak?
Yeah, that's a great question as well. So if we think about the brain, where, you know, the neurons that you have at birth are going to be the same neurons that you have in your 8th, 9th decade, right? They're the same ones. Neurons don't reproduce, they don't replicate, they are non-dividing cells. So to maintain the health of the neuron, everything else has to be recycled the whole time, right? So the mitochondria are constantly being made, biogenesis it's called, and cleared through one of the pathways I showed you. So it's really important then that we remember that that cycle of making more and clearing can be modulated even when the neuron is 50 years old or 80 years old. So while the neuron is still alive, those mechanisms can be harnessed. Remembering that the condition and all the biological processes are a little bit asynchronous. So we don't have this point where every single mitochondria is damaged. What we have is a sort of, some mitochondria are less functioning, some are more irreversibly damaged, others are reversibly damaged and can be repaired. And so we've got a kind of heterogeneous group of mitochondria at any one point before a neuron dies. So really we'd be saying that every stage of the condition, you should be able then to make better mitochondria and to remove the non-functioning mitochondria. And I think a lot of the work that we've done have suggested everyone worried that it was just a late stage process. And I guess we're saying, no, we think it's early actually in the condition based on the model systems we've looked at. So actually, I suspect that boosting mitochondria at any point in the condition will be helpful.
Thank you very much.
So
it's never too late.
That's based on our data.
So the additional question from Stephen, he was wondering whether you can say whether non-motor symptoms in PD are more related to mitochondrial health than the motor symptoms, or is it difficult to make a difference between both?
Yeah, I mean, yeah, it's a great question. Of course, what I showed you at the beginning were those dopaminergic neurons that I think are more vulnerable, and they're the ones that account for the motor symptoms. So is that why they're affected early or mid-disease? But we do see mitochondrial dysfunction in the cortical neurons that are involved in cognition as well, right? So, and that's a non-motor phenomenon. So I think I think the same processes are happening in multiple neuronal subtypes, but some are more vulnerable than others. So the motor ones are possibly more vulnerable than the non-motor ones, but the non-motor ones also have mitochondrial dysfunction in them, at least in the models.
Thank you, Sonia. So let's kill again two birds with one stone. Robert and someone anonymously have the same question about dietary changes. I know we touched on that. Is there anything you can tell us? I know it's maybe not the angle of your research so much, but actually, is there anything we can do to take care of our mitochondrial health through what we put in our bodies every day?
So, I mean, I think that, again, you know, we use the cardiovascular and the diabetes literature to tell us, to guide us really, because we know that the Mediterranean diet, and we know that, you know, trying to maintain the balance and a very kind of protein-rich and a vegetable-rich diet is healthy for us. And it's healthy for us in many, many ways, of which I'm sure mitochondria play into it, but it's healthy for us peripherally in the rest of our body. But that will have an effect on the brain. What I can't tell you is the right diet for the brain directly. I can only tell you what I think that diet is for the rest of your health.
Thank you. I just wanted to share, it's not a question, it's a very helpful message here from Jerry, who basically tells us that NAC, there is an NAC clinical trial in Philadelphia at the moment, as looking at NAC as a precursor for glutathione. So that's exactly what you're talking about. So that's maybe one we can look at afterwards. Then interestingly, there is someone called the BST Club, which I bet you is the broccoli seeds something club, because they are asking you if you are aware of the effects of sulforaphan on opening the NRF2 pathway to relieve ROS. I don't know what ROS is, but here we go.
Well, I'm not aware of it. So I'm not going to comment because I don't want to comment on something I don't know enough about. But I do think the NRF pathway is a good pathway to study and to manipulate.
So Robert is asking me, this question for me here, to repeat the name of the naturopath that I mentioned. She's a lady called Laurie Mishley, M-I-C-H-L-E-Y. She's very famous as a naturopath, and there is tons of things about her on the internet. And you can find a great presentation by her a year ago, I think, on our YouTube channel, Laurie Mishley. Yes, Richard is asking, sorry, we have 9 questions actually, and some of them can be quite quick. So we're getting to the end of it. I'm going to try to wrap up. Richard is asking whether he came across an announcement that a blood test can now detect if Parkinson's is present. Now, actually, I've seen several announcements. There probably are 3 or 4 of them out there. And he's asking how reliable this test would be. I don't know whether your angle of approach to the condition gives you an opinion about this.
Well, so there's been a number of blood-based biomarker tests in the last year or so. I think the one you might be referring to is the most recent one that came out of a combination. I think it's 8 different proteins and a sort of machine learning style algorithm that makes a prediction on the likelihood of having the condition. I think the only thing to say about the ones in blood at the moment is that they've only been done on research cohorts. So groups of participants who are within a cohort that's been deeply phenotyped or examined by doctors, and their condition is really understood and well known. And until we do the real world trial, which is taking the test out, will we even begin to understand how well it functions? So I think it's really promising. It's a really promising field that we might get a diagnostic for Parkinson's in the blood, not in the cerebrospinal fluid, which is becoming apparent. But the blood-based ones are still a little bit early and haven't left the research setting yet.
Thank you. Peter has an interesting question about levodopa-induced dyskinesia, and he's asking whether it means that the cells have been damaged by levodopa. So our previous speaker is someone called Jonathan Zachner-Bronstein, who is slightly unusual in what he says about levodopa, and basically saying that actually there might be too much levodopa at the intracellular level, which is not always measured as regularly as it is normally. I don't know if you have a view about the potential role of excessive levodopa or long-term levodopa on mitochondria and the cells in general.
You know, I'm not aware of the literature studying levodopa on mitochondrial function, but we'd always understood that levodopa-induced dyskinesias was related to the existing neurons upregulating their receptors and becoming supersensitive to the levodopa. But you know, again, this is old literature, so I'm not sure I know enough to be able to comment on that.
Okay, I think we will wrap up in the next few minutes to close down after an hour and a half, which is a lot of your time. So thank you very much, Sonia. I don't know if you have heard of that protocol by an Italian neurologist called high doses B1, vitamins B1. If you're not familiar with it, then that question will not resonate, but because someone is asking about the potential impact on mitochondrial function. So I will pass that one then.
Thank you.
We have a presentation on that topic, as we do seem to have on everything on our YouTube channel again.
Great.
By someone who wrote a book about it.
Much better than my response then.
And then I will just keep maybe that last question here from Talib, who is asking if diabetes is a risk factor for PD, because I know you've been looking at that quite a bit. Yeah. And again, the same question about what would be the dietary regime that would help. So sorry, that is something that comes back as a theme.
Yeah, so we think that people with diabetes have an increased risk of Parkinson's. And we think that people who develop both conditions, their Parkinson's can progress more. And there's also pretty good epidemiological data which suggests that people who are on certain classes of anti-diabetic drugs have lower risk of Parkinson's. So this interaction between diabetes, insulin resistance and weight in the periphery to what is happening in the brain there is a clear relationship. And we're doing quite a lot of research to try and understand that relationship, but it's not very well understood. So insulin resistance in the brain operates through a slightly different mechanism. So anything that reduces the diabetes, exercise, of course, weight loss, and the diets that are again suitable for diabetic patients, we think should therefore also improve the group of patients who has Parkinson's because of insulin resistance in their brain. Not proven, but that's where we think that's where we think the link is. So there is definitely a link. But again, what we're not sure of is that all patients, or is that subgroups of patients? Is there a subgroup of Parkinson's patients whose link with diabetes is stronger than others? And I don't know the answer to that yet. But I think it is absolutely fascinating and also hopeful that these anti-diabetic approaches are useful in Parkinson's.
Sonia, I will stop it there. We have answered close to 40 questions, which is a very solid number. I don't think you expected that. And I hope you enjoyed them as much as I did. Thank you very much for coming back to us today, because I think you're freshly back from holiday, so it's a very long day for you, and for making yourself available. But more importantly, for the time you dedicate to helping people from a clinical standpoint and from a research standpoint. So thank you so much.
Well, thank you so much for having me. And I'm sorry I'm not able to answer everything, but I tried
No,
to
no, no, no,
the
but
best of my
you have
knowledge.
answered plenty. And actually, and of course, our audience is very varied, and the questions come from all directions. I just wanted to say before I let everyone go that the next session will be taking place in September. There is not a date yet, but in September. And we will have the pleasure of listening to Professor Philip Schieperians, who is one of the world specialists in gut, brain, if you want, link in Parkinson's. And he will talk to us about the gut microbiome and PD. So for all of you who had questions about dietary recommendations, I will tell him in advance that you should expect a lot of questions about that. And so thank you, everyone. Have a great month of August. Enjoy the sun and the holidays. And I will see everyone in September. Thank you, Sonia.
Bye-bye.
Bye-bye.